we'll be showing you how to go from raw sequencing data to confident antibody leads. And we'll also be sharing practical workflows on how to streamline lead prioritization. So to introduce the team, my name is Monica. I'm the head of go-to-market at My Laboratories, and I will be your host today. We're also joined by Sam Poslavsky, the CEO and co-founder of My Laboratories. And we also have Sophia and Maddie, who you might be familiar with, who will be monitoring the chat today. Just a reminder, today's session will be recorded, and you can also drop your questions anytime directly in the chat or the Q&A, and we will have time to answer them at the end. On behalf of the MyLab team, thank you guys for taking the time to be here. We know how busy discovery teams are right now, so especially in today's fast-moving therapeutic landscape. Let's get started, and I'm excited to pass it over to Stan, who will talk about today's topic.
Thank you very much, Monica, and thank you very much for joining this masterclass. So just to remind, today's focus is in vitro antibody discovery, which typically involve pinning rounds with display technologies. So that will be our primary target for today masterclass. We'll have a separate webinar for in vivo antibody discovery next week. So if you're for in vivo, then you can also check our schedule. So the typical workflow of in vitro antibody discovery involves display libraries and pinning grounds. Today, we are able to generate tens of millions of sequences through those campaigns, and the actual bottleneck comes to the analysis part when we have millions of those sequences derived from NGS sequencing, and then we have to get our high quality leads. Why this is a bottleneck? Well, the standard way how we approach to the lead selection, or like the old way how we approach to the lead selection when we have display libraries, is just picking the top abundant candidates. And this actually has problems because we're picking the top N candidates from our campaigns. We may have biological biases. The top hits are often maybe just fast growers or like artifacts and not the best binders. Yeah, or another set of problems that if we just blindly select the top leads is that the leads that we're selecting may be fragile and, for example, expose different liabilities and fail downstream. Yeah, and if we don't have backups, then the entire campaign may fail. And finally, with just picking the top candidates, we're actually losing the whole diversity of the repertoire and miss the diversity. So that's why we use NGS, and in the modern campaigns, we use and rely on the NGS. It allows to rescue rare gems and identify high affinity binders hidden by slow growth, for example, have a backup. So if we have candidates that fail downstream in developability assays, then we have backups that we can use to secure our campaign and de-risk early with the in silico methods that allow us to predict developability properties early. We can better navigate through selection. But this deep mining actually requires pretty sophisticated computational workflow, and that's what we will be focusing on today, how to build and engineer a campaign to have at the end de-risked lead panel of candidates. So our plan for today is to use some real project, pretty complex project. It's a VHH nanobodies, although our platform supports all other modalities of the data, including single chain, variable fragment, single cell, whatever. Yeah, but just for example, today we're using VHH. We will have a live demo going step by step from end to end, from the row reads to verified leads, yeah, using our methodology. And at the end of the session, you will have time to ask your questions. And please, if you have questions, share them right now. Yeah. So you will learn how to escape the abundance trap, and instead of picking the top candidates, prioritize families over the individual fast growers, de-risk in silico your lead panel before you waste time on the synthesis, and bridge the wet lab by importing for assay data, immunoassay data into your NGS and map this onto NGS profiles to uncover the full potential of your campaign and of your data. Please stay until the end, because at the end we will reveal a special bonus for the attendees. So with this in mind, let's start how we will do this, how we'll engineer this, the risk lead panel. We'll use Platforma. That's our end-to-end antibody discovery software, which integrates sequencing data, functional validation, developability, in silico assays, etc. So with Platforma, we have an end-to-end solution, starting from the raw data to the lead selection. And we will go through three parts: deep mining, including the clone typing and annotation of our antibodies, multidimensional filtering to build our lead panel, and integrating wet lab data. Right now, Monica, I think you wanted to post a poll. Yeah. So
Exactly. So right now we're talking about the analysis portion of the discovery. So I'm going to launch our first poll, which is what software are you using for antibody discovery?
yeah, well, thank you, Monica. Please reply. This will be super helpful for us. The data set that we're going to use as a demonstration is pretty complex VHH data set. It's a real library, although it's a synthetic library of VHH with high diversity. So the backbones of the nanobodies were used from the real alpaca, and it's a real therapeutic campaign. There were two rounds. There were performed two rounds of selection against a difficult target, and a very important thing, the crucial thing that we always recommend to antibody discovery people is to use negative selection arm or negative control. In this case, it was just a beads-only selection to occasional specific binders, and that's, as I will show you, is extremely useful and extremely important to have this negative selection arm to get real leads. It's MiXCR, so it's around 10 million of reads in total, and the data has sticky binders, aggregates, and different artifacts. So what we'll have to do today to build our diversified and de-risked lead panel is to navigate through different traps. Yeah, like the trap that the top enriched clone may be just a sticky binder, yeah, or even if we're selecting the real specific binders, they may fall with the developability risks, and that's we will have to de-risk and understand and filter early. And the real gold, the real hidden gems, maybe not that like fast growing that fast, yeah, or be looking very nice, but at the end of the day they turn to be specific, liability free, and validated by the wet lab assay data validation. So with this, let's start. Let me share my screen one more time, and to show you the software. So here we have Platforma software, just one short paragraph. What you're seeing is the graphical user interface. It's a very powerful user interface which is designed for biology users, but it's very bioinformatics friendly. It's kind of white box. So all the things that I will be showing you today are open source. All of the algorithms and everything is very transparent for bioinformatics and computational people. At the same time, user friendly for the biologists. So that's the place where everyone can collaborate and go deep into the biology. So as I explained, the data that we have here today is here. So we have a couple samples coming from the screening library. There are two rounds of panning, round one and round two, yeah, against the target. That's one pathway that we have here. And we also have a negative control arm, which is just selection with the beads themselves. And here, as in the very, very common case, we have just the last round of enrichment for the negative control data. So the workflow that you see here in this project on the left side is pretty straightforward. We are starting with annotation and clonotyping, which is powered by MiXCR. It's a gold standard tool that we developed almost 10 years ago. It corrects for PCR and sequencing errors if we have barcode like UMIs, not in the case of this data, but if you have UMIs that process the UMIs, and there's a lot of quality checks, so very standard thing. And in general, if we look on the repertoires generated through this campaign, through rounds of enrichment, it will look very, very nice. So for example, if we look on the rarefaction curves, then we will see that we have in the screening library pretty high diversity. It's growing, so we are not sequencing to the full depths, but we are more or less fine. With our painting grounds, we have saturation, yeah. So we have funneling, which is also very good. If we look on the diversity, for example, of our libraries and on the dominance, like Gini index, we will see that from screen library towards round 1 and round 2, we have the increase of the dominance, and we have a real funneling effect. We have growers, so everything looks nice with the data. We can look on other properties of the repertoires themselves. But now let's go to the very end of this workflow, to the place where we're selecting leads. Imagine if we are selecting leads and antibody candidates from this data using Excel. What we will do? Yeah, I'm going to do this live here in our antibody TCR leads block. So we pick the top N candidates. Let's say 100 top antibodies through the campaign. So meaning that we're prioritizing by the abundance, which is in our case mean fraction of rates. Yeah, and let's say we select 100 antibody candidates just by the abundance. So that's what I did here for the beginning. And with this, we are getting 100 of top antibody leads. Yeah, I will explain these things through our process of escaping traps in more details. But if we just look on those candidates on the, for example, multiple sequence alignment, we'll see that we have some diversity here. We have some candidates that are really diverse. So we see the diversity in the CR3. Yeah, and if we group, for example, by the similarity, we will see that there are certain families, so there are certain groups which have very similar sequences, which is actually what we want to avoid, and we will avoid this in future. But all in all, this looks fine, and in a old way, we can just use those leads for the downstream synthesis. But before doing this, let's ask one question. Since we have a negative control, what's the fraction of those high abundant candidates that we picked here with the standard top and picking are actually not real binders, but sticky antibodies? That can be done really easily with Platforma. Let me do this live. So I am in a block which is called Graph Maker. It's kind of Prism graph pad embedded in the platform where you can build really sophisticated and very advanced publication-ready visuals. So let me add Graph Maker here, and let's compare. So here we selected 100 leads out of hundreds of thousands of millions of our antibodies from our GS data, and let's compare. Let's use stacked bar plus stream area chart to see how let me see how many of the candidates that we selected are actually belong to the negative control. So here we have a negative control sample as one of the paths. So let's group our candidates by path. On the y-axis, we are showing the read fraction. Let's filter this to the selected leads. Yeah, and now we have read fraction here. We have negative control target, and let's, for example, use CDR3 as a secondary grouping here. Let's also use mean here. Let's make it larger. Let's make it nice. Let's remove legend. Let's use different color. I'm doing this really fast, but since I'm very familiar with the platform, so my goal here is just to show you what we have. So here we're looking on the selected leads, on the candidates that we selected by a very simple filter, top N candidates. And now we're comparing the abundance of those candidates in the negative control samples, yeah, without the target, and the target samples. And we see that many of the leads that we selected here by default, yeah, they are present in the negative control. So this is a very typical situation. So we always have, depends on the library, depends on the experiment, but if we have this, yeah, picture here, it's pretty typical. And that's very bad, meaning that we are selecting just sticky binders. What we want to do, we want to get rid of them. We see that their abundance in the negative control is really large. Yeah, so this is an example of the trap that we have when we're picking top and candidates blindly. Yeah, another thing that we can observe here is that some of the candidates, especially if we, for example, add Kabat numbering and compare them on Kabat numbering or just multiple sequence alignment that we already did here, they look pretty similar. So the diversity is not that great on the, and we have really families of similar antibody candidates picking here, meaning that if we test them downstream, we will actually test the more or less same profiles. So how we can escape those basic things? Those basic things we can escape using the enrichment analysis. And for enrichment analysis, we have a clone type enrichment block in the platform, yeah, which is like really straightforward bioinformatic algorithm under the hood, very transparent and open source, that calculates the log-to-fold change enrichment scores across rounds of enrichment that we have. We have our initial library, we have round 1, round 2. We also have negative control, so we can configure here the negative control path. So we select that we have negative control here. And one other crucial thing is that we can configure this enrichment analysis to calculate the enrichment factor and to track the enrichment, not for individual clonotypes, individual antibodies, but for families of clones. How we do this? We use clonotype clustering. Clonotype clustering is based on the MMCQS tool. Yeah, we cluster our antibodies by the full VDG region sequence. We can define parameters. There is documentation. There are some defaults that is good to use for the in vitro campaign. We always recommend to cluster by the full VDG region sequence. In case of nanobodies, in case of a single chain variable fragments, then you will use here the full SAFE sequence, etc., etc. And with the clustering, we are able to group our antibodies into the families with more or less similar sequence. The parameters here are actually pretty important. So since we're doing in vitro campaign and we have the full length with G-region sequence, we put the pretty high threshold for the minimal identity, 95%, meaning that we allow difference in 5% of the proteins in the cluster for the members of the cluster. And for different modalities, again, for example, for SAFE, we would use like different number here, or as possibilities that we can use depending on the specific campaign that we are running. We can run the clustering not by the full VDG region, but for example, by just CDRs or just CDRs. It's all possible here to configure. But once we did the clustering and grouped our antibodies and families, we can run enrichment analysis on the clusters. And that's crucial because for the reasons that we will explore below. So once we run it, enrichment, we can check, for example, frequency line plot to see how our clusters, let me change the color to one or like this one more, we will see here those are individual clusters or families of the antibodies and their abundance and their like frequency change, log to frequency change through rounds of enrichment. Since we have control, we can also check whether we have how many of our enriched candidates from the target pathway are actually belong to the negative control, and things like that. So we can really look in different ways on the clusters and individual antibodies that are enriched through rounds of selection. What this block also does, it assigns the enrichment quality to each of the families, to each of the clusters that we have here. And those enrichment qualities that we have here assigned, there is a documentation how and based on which criteria those values, weak binder, stable binder, non-binder, parasite that we have here. What are the parameters, how they are assigned. And basically, they are very, very transparent and configurable by the user. But once we run it, we now have the enrichment quality. What we can do now, we can select our antibodies not just by the top abundance, but, for example, let me configure this. we can select our antibodies by the enrichment score. So, for example, we want to select only the candidates that belong to the families which are either stable binders or rescuers, meaning that their log2-fold change score across rounds of selection satisfy the criteria that we define in the enrichment analysis. We're selecting now only the candidates that are antigen specific, and we are dropping all of the candidates that are enriched in our negative control. Yeah, so we are looking here only for the antigen specific, and as you see, negative control is excluded here, so we're selecting only the real antibodies that bind to the target. And we can impose the log2-fold change threshold. The value generated automatically based on the profile of our library. Yeah, but we can modify this. The other crucial thing that we do, those numbers are calculated for families. So for each cluster that we did with the clone type class rank, for each cluster, we have this enrichment score assigned. Yeah, so here we're selecting those clusters. And now we want to pick the candidates, the antibodies from different clusters to diversify our lead panel. So here in the diversify by, we can select the clusters that we have in our data and diversify our leads by picking clones from different clusters. So we first filter by the clusters. For each cluster, we calculated the enrichment quality, the log2-fold change. We dropped those clusters that also enriched in the negative control, which are just sticky binders. Yeah, and now from those clusters, we're picking the candidates. We're prioritizing those candidates again by starting with the top performing clusters, with the clusters with the max log2-fold change. And then within the cluster, we can prioritize in different way. For example, we can prioritize again just by the abundance, like the mean fraction of reads. That's the fraction of reads supporting the antibody as the ranking score to pick the ones that are more abundant. Or we can select, for example, the ones that are closest to the centroid of the cluster or more distant from the centroid of the cluster. And we can tune those parameters in different way, depending on the specifics of our antibody discovery campaign and our understanding of the underlying biology. Here, I'm using the defaults. So we're just looking on the enrichment score, we are diversifying by picking from different clusters, and within the clusters, we are ranking by the fraction of reads, meaning by the abundance. So let me run it here in a live way, hoping that it will work nicely. Okay, it's loading. Yeah, done. So we rebuilt our lead panel using those criteria, using the enrichment scores and the diversification with the clusters. Now our lead panel should be better. And at least the top abundance trap is the thing that we should avoid. Let's look back on our visual where we compare the amount, the proportion, the presence, where we check the presence of our selected leads in the negative control. And you see that it changed. Now the leads that we have, they are really enriched in the target samples, in the pinning grounds against the target, and absent in the negative control. Very good, very well. Yeah, but that's not the end. If we look further, the question is, are those candidates are developable? Yeah, that's the second trap we want to avoid as soon as possible to not waste time and money on synthesizing the bad candidates. And again, this can be done here in the platform. We have an antibody sequence liability block. So basically, this is the block where you can configure which sequence liabilities you want to check in silico on the whole data set. Yeah, so here is the list of the defaults that we have. Yeah, and different liabilities that we sequence here. We are talking about sequence-based liabilities and very well-known motifs in the CDRs that can fail, may make antibody failing downstream. Yeah, so those liabilities are calculated here for the entire repertoire that we have in the data set. And if we go to the antibody TCR leads, let me just show this column, liability risk and sequence liability score. Like, let me make them visual and move to the top here, sequence liability summary, and I think liability risk. Let me just move it to the top to make it visual. As you see, there are many, many properties for the repertoire that we have here. So now we have our leads. For our leads, we have the many different properties, but looking on the sequence liability risk and sequence liability profile, we have many of the candidates that expose a high, that we just selected by default, looking on the enrichment, diversifying by the clusters, a very natural procedure. We see that many of them have, well, different liabilities that are pretty critical for our lead selection. And if we just, again, blindly pick those leads, we may have a substantial portion of them failing downstream in our assay analysis. So that's what we have here. Yeah, and actually, if we go to clonotype space, that's a very, very useful and powerful instrument that we have on the platform, allowing to visualize the entire space of our antibodies. Yeah, super important thing, by the way, because it's super useful when you're iterating through the parameters, when you're testing what parameters work best for your data. So here, each dot is one antibody candidate, is one antibody from our entire data set. It's a UMAP plot calculated based on the sequence. So the idea of this visualization is that two antibodies located closer to each other if they share similar properties, whereas the similarity is defined by the sequence, by the protein sequence of the antibody. And we see that there are some clusters of similar antibodies. We can use LASSA tool, for example, to compare those antibodies on the multiple sequence alignment, et cetera, et cetera. But what most important here that we can layer all the information like enrichment scores, liability profiles, and all the information that we have on this UMAP to interactively see what we have in general in our space. Like, for example, yeah, liabilities. Liabilities is what we want to avoid. So if we look only on the sequences that have high liabilities, a high risk, I just highlighted them here on the plot. Yeah. 1, 2, 1, 2, 3, 1, 2, 1, 2, 3. There are too many of them. Let's highlight those that do not have liabilities. Yeah, that's better. So you see, I highlighted only those clones, only those antibodies that do not have any liabilities. And you see that our entire space is not that great in that sense. So that's some signal to our screening library. In the case of this specific experiment, that's the reason, because the library was generated with not in the very efficient way. Yeah, and that's typically what you want to avoid. But although that's what we have here, and that's a reality. Yeah, so we have the highlighted antibodies are those that are like do not have critical liabilities that we can't fix downstream. Let's also layer here, for example, the enrichment quality as the grouping. Yeah, so just to see, yeah, we have colored. Let me change the color to make it more colorful. Yeah, we colored our entire space of the antibodies by the enrichment quality, which is calculated in our enrichment block using the log2fold change. So basically here, let me just explain. The stable binder is the antibody which shows pretty stable increase of the abundance through rounds of selection. So the log2fold change is stably high across the rounds. Yeah, weak binder is obviously don't show this, and the non-binder is the ones that just don't show any substantial. Well, show some some growth in the local fold change, but not substantial one. Yeah, and we see that the amount of stable binders that we have is in the intersection of highlighted antibodies and the ones that are red. Yeah, and what we see here that, yeah, you see here, many of our stable binders have high liability risk. Yeah, here we have some stable binders that don't have any risk, and which is nice. So from here we will be picking the good antibodies to build our the risk lead panel. But what we also see that there are some weak binders. If we zoom in closely, we will see some weak binders or that actually liability free. So we have something here. Other thing that we can look is, for example, the give me a sec. We want to see how many of our candidates are non-specific. Yeah. So I'm coloring by the binding specificity. Binding specificity is what is also calculated in the enrichment block. There are two values of binding specificity. The specific, antigen specific, meaning they are enriched against the target, and the negative controls, just non-specific. And what we see here that the substantial part of our entire space, actually, we have this negative binders here, which are actually also do not have any liabilities. Yeah, here, etc. So we have pretty many in this specific library, in this specific campaign. We have pretty substantial number of false binders in the data. So this clone type space is a very powerful instrument allowing to explore the entire space of our repertoire and see what we have in general. But now returning to the liabilities, to our antibody TCR leads block, since we have pretty high number of candidates that we picked, even using the enrichment, analysis, negative control, diversification with a high risk of liabilities, we can configure now the final filter that we would use in this campaign is liability risk. And let's say we don't want to have high risk candidates with a high risk of non-developability. Yeah, so we are excluding only high risk candidates, and the others, let's say, we can fix. Yeah, what also important that probably when we're selecting when we see that we have many of antibodies exposing the developability risk in the data, we may lower the default threshold that we have for the log to fold change. Yeah, and now let's run this analysis. So with this configuration, we're selecting antibodies that don't have any liabilities, sequence-based liabilities, that are enriched, that are enriched specifically to the antigen that we're using in this campaign. We diversified this lead panel using the clustering, and we picked the most abundant candidates that are still liability-free from those clusters. And with this, we will have pretty good results. Maybe not that much, and it depends on the diversity of the initial library and biases that we have in the initial library. But still, we have pretty good number of candidates. Now we have only the candidates that are free of liabilities. We diversified them using the clustering and selected them through the ranking score that we use here. We may use different of them. So, for example, let's put this way: if we see that in this campaign we have pretty some number of candidates that belong to the same clusters, meaning that we don't have enough diversity among the candidates that really bind. Yeah, we may diversify further within the cluster by ranking the candidates and picking them as farmost candidates from the centroid of the cluster. Yeah, and then will give us better result in terms of the diversity. Then if we look, let's run it. should be pretty fast. Yeah, so once it finished, if we go back to the clonotype space right here in the antibody TCR list block, we can see how the picked candidates, how the specific selected leads located in our entire clonotype space. Yeah, and let me make this bigger, and we'll see that depending on the parameters that we set and actually what we want to achieve, we have candidates located in different clusters in different parts of our entire antibody space. Let me just highlight this in a slightly different way. Yeah, let me use it as a size to see where our candidates picked from the entire space. And actually, what we want to avoid, and that's the next step of our iteration, we have pretty many candidates located from in this specific cluster, which will not have sufficient diversity. If we, for example, pick them here with a multiple sequence alignment and compare, that's not enough diversity in the candidates that we're picking here, and that's what we want to avoid further, which we again can do. But I don't want to eat all of the time by using different prioritizations and runs here. Before doing this, I want to show the last piece of this puzzle is how we can close the loop with the wet lab data and wet lab validation of those candidates. So when we select and pick some of the leads here and test them in the ELISA assays, we can import back the ELISA results with the immunoassay data block. So, for example, here I have 6 candidates that were picked here in this campaign and then tested with the ELISA assay. Yeah, and we can import this assay data in the FASTA form of just CSV or TSV or Excel format back and map to the NGS data. So here are the leads that were tested with the assay, functional assay. We have some parameters that were assigned based on the immunoassay data. The most important one is the status. So we checked which of those leads are actually fail in the functional assay or which pass our functional assay validation, specific binders or no expression or no binding or like some polyreactivity. So we can make those notes and import this information and map this information back to our entire NGS space of candidates. Let's say here we have looking on our family of candidates that pass our ELISA functional validation. We have pretty substantial families of like 100 of antibodies. That's pretty large family of antibodies that we assume, based on the representative that we used here with functional validation, are also good enough, and we can use them in our lead selection. Let's just, for example, move down the clone type space under the immunoassay data, and 1, 2, 1, 2, 3. Oops! I rerun it, so it will take time to reload. Status notes. Let me use the status notes from the assay data as the grouping color to see where, well, it will take time because I clicked run button suddenly and it will take time to reload. But the idea is that we can import the functional assay data. Well, it takes time because it runs against the entire NGS data set, the matching algorithm. And we can import this assay data and highlight, well, yeah, it loaded here, and highlight where on the entire clone type space located the families that we validated with the functional assay data. So for example, here, as the status from the assay data, we see where our like families of and the largest one actually here that we have is just the failing candidates. Yeah, so our passing candidates, let's highlight them. Let's highlight them that pass. There are not so many on the entire space, but here they are. They are located here in one specific place, and here in 2 specific clusters of our entire space. We have our families validated with the functional assay data that we can further use in the TCR data selection. So, for example, if we did enough, for example, historical testing. And we can import all this information in the assay data block and then configure here as a filtering or ranks. Let's say we can filter only for those families that are passing our assay validation. And with this, we'll have enough backup variants or just better prioritization. Yeah. So we first will fill better to use actually as a ranking. We first will pick the candidates that are belong to the families that we validated with a functional assay, and then the rest of them. So I hope I was not too chaotic, but within this 20 minutes, and was able to show you the power of the platform that you have under your fingerprints, allowing to prioritize and select the antibody candidates, navigating through the traps in our display campaigns, and reliably build the diversified and de-risked lead panel. One thing I didn't mention here is that, of course, the antibody discovery campaigns are complex, and navigating through all those things requires iterations and computing power. And this platform works at the scale of millions, dozens of millions, hundreds of millions, and billions of reads. And you can process tremendous amount of data in the same way as I was processing this data set quickly and interactively and transparently. All of the algorithms that are here, they are open source and available with pretty rigorous and detailed documentation on our portal. So with this, let me go back to the slides. Yeah. And the few things that I want to mention before going to the final, that I didn't show today some of the important things that we're building on the antibody front and already have in the beta version. It's structural modeling. And for example, ability to predict liabilities, not just based on the sequences, but based on the structures of the antibodies, and use it in the same way as we are using sequence liabilities to discard candidates that we can confidently say will not be developable, and the 3D visualization of those structures to project different properties and view nicely the 3D structure of the antibody with different modalities. We also bring in the peptide discovery, where the same entire workflow that I showed for the antibodies with the lead selection will be available for peptides. And in Q2, we'll have dedicated modules for antibody humanization. So that's basically what I wanted to share today. And Monica will post the next poll in the Zoom chat regarding what. Monica, please.
Exactly. So at my laboratories, we're always trying to build what we're working on, what people are needing. So your feedback really informs us what to prioritize. And so we want to ask, and you can choose multiple, or you can choose a single answer based on the roadmap. What would be the most relevant to you?
Yeah. So one thing to mention, that you can download the platform right now from our Platforma website. So all of the workflows and all the algorithms that I was showing today are available. We have technical notes, application notes, we have online documentation portal. All of the algorithms and bioinformatic tools that are used are very well known, published, and open source. You can also run the platform on premise, so on your infrastructure where the data do not leave your closed computational environment, or in our cloud. And for the platform is free for academic research. That's part of our long mission. We provide all our software for free for fundamental academic research. And for industry, we have a special offer for those who attended the webinars. You have a 7-day free access to our cloud servers to explore the exact same projects for In-Vitro and other projects that we have on the platform online. or you can request a 30 days on-premise trial where you install the software in your infrastructure and run all the analysis there.
Thank you so much, Sam. So the QR code on the screen is directly to book a meeting with us. That way, our bioinformatics team can assess your workflows and also recommend some ways that you can optimize with Platforma, as seen with the things that Stan has showed you today. So now we're going to move over to the Q&A. So we have a question from David. Do you find that you need to do a parallel phage display selections to increase the significance of your enriched clone ranking?
Yeah, it's a very good question. And there are two answers. One answer, yes, it will increase statistical significance. And for some challenging campaigns where you don't have enough, that nice enrichment and things are not going well, it may improve the results. But so far, in many, many campaigns that we saw in industry and academia, what really important is just negative control. But then having replicates or parallel enrichments does not give like much to add when you come down to the final lip pen. But negative control is essential, super important. With replicates, actually, we have what you can do with replicates. If you run multiple, let me share back my screen. If you run multiple campaigns in parallel, then you actually have, or just have real biological replicates. Then what you can do instead of just basic enrichments or and log-to-fold change, you can do a differential abundance analysis. I'm showing you some other project that we have on the platform. So it's a different, it's TCR discovery, but gives like an idea. You can run the differential abundance analysis to find what are those antibodies that are differentially more abundant or differentially enriched with a p-value, yeah, among on top of just log-to-fold change through your campaigns, and this gives much more statistical power, of course. Yeah, it's very nice. But as I said, for just standard display campaigns, it's very rare when people do this.
Okay, great. And then we have another question from Eileen Burke talking about how does the software trace sequences between panning rounds? Is this UMI dependent? So one thing, as we mentioned, Platforma is an end-to-end platform, so you never need to leave from data to lead. And we didn't have much time to cover this, but Stan can go through and show you how to upload data.
Yeah, so that's a great question. So there is a clone type definition. Basically, the clone type enrichment block is a super straightforward. You have clonotypes, and you have abundance of each clonotype in each sample. Yeah, we have samples like sequencing samples. In each sample, we have a clonotype. How we define the clonotype? In case of in vitro antibody discovery, we define the clonotype for nanobodies specifically by the VDJ region, by the protein sequence of the VDJ region. Yeah, so the clonotype is just a protein sequence of the VDJ region. That's the definition of clonotype. And then we have abundance of each clonotype. I'm showing you a clonotype browser where you can browse your samples and the content of each sample. So here are the samples. Yeah, in each sample we have clonotypes. Those clonotypes are defined by their full VDJ region sequence. They have CDRs, frameworks, etc., and the abundance of the clonotype in sample. This abundance, dependent on whether you have UMIs or you don't have UMIs, is measured in the number of reads or number of UMIs. Like here in this specific project, it's number of reads because there are no UMIs in the data. But if you have UMIs, and we fully support UMIs, and the MiXCR clonotyping, the tool that annotates, aligns, and corrects for PCR and sequence errors, you can configure to have let me just show you, for example, generic amplicon. You can configure the where it is. UMI, some generic amplicon with UMI. So we will support all types of libraries, all types of sequencing. all types of chemistry. All in all, you can put the parameters of your UMI and tag pattern of your UMI. Let me show you another example. Just it will be clear. Right in the settings of the block. Let me show you. Like we have MiXCR amplicon alignment for fully synthetic libraries. Yeah, and where you could just put the scaffolds, the backbones of your synthetic libraries as a FASTA parameter, and then specify the read structure like tag pattern. Yeah, and put the regex for where you have UMIs. Yeah. So once we have defined clonotypes, then the tracking of the clone types across rounds of enrichment is done just by the sequence, by the protein sequence. Now we have the protein sequence of the VG region, and we know its abundance in each of the sample. That's how we connect the dots. Yeah. Next question. If anything is not clear, you can always reach us after the webinar. So next
Exactly.
question.
So, Stan, let's talk about the one that's about defining regions for clusters other than CDRs or framework first.
Sure, good question. So here it depends on the library. So for this specific library, it's nanobodies. Yeah, for nanobodies, we can define like CDRs and frameworks, and there are also advanced settings to if we select, let's say, just for example, if we select CDR3, then we can redefine the way how we define CDR3 and trim some nucleic amino acids from the start or end of CDR3. We can combine multiple of them, so we can use we can use combinations here. If we run single chain variable fragment, then you will have both heavy and light here. If you run single cell analysis with standard genomics, for example, you will be able to select heavy and light here as well.
Great.
Monica,
And
I
final,
don't see the question, but I hope I answered.
yeah, I answered it. So the other question, also from her, is about negative control selections from round one. So the two streams are never mixed in one selection.
Yeah, so here specifically in this specific study, yes. So it's just two completely separate pathways. So one page we started with the same initial screening library, and then one pathway is the enrichment against the antigen target, and the other pathway completely separate. They were not mixed as from the negative control, but both started with the same initial screening library.
Great. And then last question. So this person usually performs antibody discovery using hybridoma and sequences the V genes of both heavy and light chains to find the best binders. And how can this be substituted using the platform? So one is if you're doing in vivo, we have another webinar next week, which I had dropped into the chat. I will just paste the link again if you were a late joiner. But I think Stan can give you a really quick demo right now.
Yeah, sure. We have a full pipeline for in vivo discovery where, well, this project is pretty advanced, but this is an example. You can do a much more basic thing with the platform. Here we have bulk data and single cell, heavy plus light data, single cell. It's all immunization campaign, and then it was sacrificed mice, sorted cells. The bulk was collected just from PBUC for a deep, really, really deep sequencing, and the sorted cells were sequenced with 10x genomics. Then we can annotate both single cell and bulk sequences. Yeah, for single cell, we are using 10x genomics chemistry, and we configure this analysis in collaboration with 10x genomics. And then with this, we have heavy and light pairs. Now with the bulk, we have the depths. And then, for example, we can combine bulk and single cell data to, let's say, reconstruct lineages. We have bulk to have enough depths and a single cell for heavy and light pairs. So we can, for example, combine bulk and single cell to reconstruct the full lineages and select the best candidates in the lineages that were constructed by the immune system of the donor automatically. We can also, and we have a powerful functionality to cluster candidates, to cluster antibodies by the paratop sequence. This allows better diversification for the in vivo campaigns when we are like constrained by the diversity of the immune system and the selection pathway it's been specific immune system to get better candidates. But that's basically the idea. And at the end of the day, we use the same antibody TCR release block, which we just configure for our in vivo selection with the parameters of in vivo analysis to pick the paired heavy and light combinations for our therapeutic candidates. I tried to do fast.
Okay, much appreciated. So, I think that's it for everyone's questions. We're still available, so you can always message us, ping us, and we'll also send up the recording afterwards. So, if you're interested and if you were a late joiner, you can the link for the materials we mentioned, as well as the link for to get your free trial. Okay, great. Thank you, everyone, and have a good rest of your day.
Thank you.